Hello everyone, my name is Elizabeth Gersang, majoring in nutrition from Medical School Diponegoro University. With this video, I'm going to explain you the effect of domestic cooking methods on protein digestibility and mineral bioaccessibility of wild harvested adult edible insect. This is based on a paper with the same title from Food Research International Journal. Let's talk about the problem. Protein, energy malnutrition, and micronutrient deficiencies, particularly iron and zinc, are still a persistent problem, especially in developing countries. One way of mitigating the problem is to improve the amount of nutrients absorbed from local diets. Let me introduce you one of the solutions. Yes, wild harvested edible insects are characterized by high protein and mineral contents with potential to contribute substantially to nutrition security. Did you know that in Zimbabwe, the consumption of insects is specific to geographical locations? Matabeleland province has the highest population of insects, while Manicaland has the lowest. Although moving worms and termites are the most popularly consumed edible insects in Zimbabwe, Eulepida species are also commonly consumed in some regions of the country. However, there is another problem. The overall content of nutrients in the food doesn't provide a full picture about their nutritional quality. The bioaccessibility of the particular nutrient is a key parameter to consider. Bioaccessibility of a nutrient is defined as a fraction that is soluble in the gastrointestinal environment and available for absorption. It is expected that bioaccessibility will vary between insect species. Most of the edible insects are subjected to some kind of processing before consumption. Processing of insects can improve quality, safety, taste, and shelf life, but can also occasionally lead to formation of anti-nutritional and/or toxic components. Boiling, steaming, frying, roasting, and drying are among the commonly used traditional methods for processing insects. There are two types of insects that we're going to talk about. First is Eulepida mashona. This insect has protein content range about 52 to 56%. And the second one is Henicus whellani, which has protein content range about 59 to 70%. For both of the insects, the iron content is about 24.2 until 52.9 milligrams per 100 grams. As for the zinc content, is about 10 to 20.9 milligrams per 100 grams. The process for both of the insects are also different. For Eulepida mashona, first we have to remove the wings, then washing, then boiling or roasting, freeze-drying, grinding, and in vitro digestion. As for Henicus whellani, what we have to do are degutting, washing, boiling and roasting, freeze-drying, grinding, and last but not least, in vitro digestion. Since this is based on a research article, so we have to talk about the material and methods for the research. First, let's talk about samples and processing treatments. For samples, we need Eulepida mashona, or hereinafter indicated as beetle, and Henicus whellani, hereinafter indicated as cricket, that were collected between December 2016 and January 2017. The varrosid and washed samples were subjected to treatments according to the following: raw, boiling 30 minutes, boiling 16 minutes, pan roasting, boiling 30 minutes followed by pan roasting for both insect species, and additional treatment for the beetles was twice boiling for 30 minutes. Approximately 100 grams insects were processed by boiling in water or dry roasting in a stainless pot. The boiling time refers to the total cooking time. The excess boiling water was drained off and was kept for the protein and mineral analysis. Processed samples were cooled in an ice bath, followed by the freeze-drying. The freeze-dried samples then coarsely grinded and milled to a fine powder using milling machine at frequency 30 for 1 minute for beetle and 30 seconds for cricket. The samples then kept at -21 degrees Celsius until further analysis. Second is in vitro digestion. Briefly, 2.5 grams of sample was mixed with 2.5 ml of milk-Q water to make a paste-like consistency, followed by adding 4 ml simulated salivary fluid, 25 microliters calcium dichloride, and 975 microliters of water. The mixture was incubated for 2 minutes at 37 degrees Celsius. The gastric and intestinal phase were performed as described. At the end of the simulated intestinal phase, the mixture was cooled in an ice bath for 15 minutes, followed by centrifugation at 4,500 rpm for 5 minutes. The sample pellet and supernatant was now frozen with liquid nitrogen, then stored at -21 degrees Celsius until further analysis for soluble mineral and protein. The third method that we're talking about is protein content determination. Samples of freeze-dried, raw, and processed insects are analyzed in duplicate for nitrogen content. The methionine used as a standard and cellulose used as a blank. The boiling water was freeze-dried first in order to determine the protein content loss during boiling. Next is in vitro protein digestibility. Protein digestibility was determined based on measuring the amount of free over amino groups quantified by O-formaldehyde method. To calculate the percent of protein digestibility, we have to divide the concentrate of free over amino groups in digested samples to concentrate of free over amino groups in total acid hydrolyzed sample, then multiply by 100. Last is mineral analysis and in vitro mineral bioaccessibility. The mineral content of freeze-dried, undigested samples, supernatant, and boiling water was analyzed by ICP-AES. To calculate the percent of mineral bioaccessibility, we have to divide the amount of mineral in the digested sample to total mineral content in undigested sample, then multiplied by 100. Finally, results. Let's talk about the protein content. First is Eulepida mashona. The results are based on 6 treatments: raw, boiled for 30 minutes, boiled for 60 minutes, boiled for 30 minutes twice, boiled then roasted, and just roasted. Twice boiling of beetles for 30 minutes resulted in a significant decrease in protein content compared to all other treatments, which is 14.66% loss. and least comes from boiled roasted treatments, which is 0.81% loss. While for Henicus whellani, there are only 5 treatments. Boiling for 60 minutes for crickets resulted in maximal loss of protein. The absolute protein loss in the crickets is 2.25 until 2.68 grams, was higher as compared to beetles, which is 0.98 to 1.81 grams. The higher loss of protein in crickets could be due to the pre-treatment before cooking. Degutting the crickets could have increased the migration of protein into water as the inner part of the insect's body exposed to water. As expected, the roasting process didn't affect the protein content. Now let's talk about the percentage in vitro protein digestibility. Overall, the digestibility of raw beetles, which is 30.6%, is comparable to that of crickets, which is 29.7%. Boiling twice for 30 minutes reduced significantly the protein digestibility for beetles, from 30.6% to 23%. Interestingly, roasting didn't have any significant effect on protein digestibility of beetles, while for crickets, both boiling and roasting reduced significantly the protein digestibility, which are 24.2 and 24.7% respectively. However, there was no significant difference in digestibility between boiled and roasted crickets. Studies in food products other than insects showed that heat treatment can be associated with either a decrease or an increase in protein digestibility. Protein digestibility of the heat treatment depends on how protein transforms during the process. For example, the formation of disulfide links in the protein matrix reduces protein digestibility. Reduced protein digestibility is also associated with interaction of proteins and phytochemicals, which form stable complexes that make proteins unavailable for enzymes. Next is iron and zinc content from fresh samples. In general, during boiling of foods, minerals are lost into the boiling water, which results in a certain decrease in the mineral content. The loss is dependent on the mineral, the boiling duration, the food matrix, and the chemical form of mineral in food. The differences in mineral loss with boiling between the two species and between minerals could be explained by the type of interactions the minerals have with the food matrix. In here, we can see the table of iron and zinc loss from Eulepida mashona based on 6 treatments. And here, we can see the table of iron and zinc content from Henicus whellani based on 5 treatments. From both of the tables, we can see that the amount of iron and zinc lost into the water was higher for beetles with maximum 0.31 milligrams and 0.21 milligrams than for crickets, with maximum 0.1 milligrams and 0.06 milligrams. For all other minerals, boiling significantly decreased the mineral content, except for copper and manganese content for beetles, and calcium, copper, and manganese content for crickets, while roasting, as expected, did not. Now we talk about bioaccessible iron and zinc from dry matter. The mineral solubility or bioaccessible mineral content gives an indication of the amount of the mineral that can be bioavailable to the human body. In here, we have the table of bioaccessible iron and zinc content from Eulepida mashona based on 6 treatments. And here we have the table of bioaccessible iron and bioaccessible zinc content for Henicus whellani based on 5 treatments. For other minerals, boiling significantly reduces the bioaccessible of iron, magnesium, phosphor, sulfur, and zinc content of beetles, but not of calcium, copper, manganese, and natrium. Roasting had no significant effect on the bioaccessible content for all the minerals except for sulfur in beetles. Boiling and roasting had no significant effect on bioaccessible iron content in crickets. Last but not least, the presence of mineral bioaccessibility. Both of the diagrams show the percentage of mineral bioaccessibility of the beetle and cricket before and after processing. Phosphorus had the highest bioaccessibility for both raw beetles, which is 88%, and crickets, 82%. Iron had the lowest bioaccessibility for both species. The iron bioaccessibility was higher for raw beetles, which is 30.7%, than for crickets, 8.11%. Similarly, zinc bioaccessibility was higher for raw beetles, which is 41.5%, than for crickets, 34.2%. The differences in mineral bioaccessibility between insect species could be related to the insect matrix. Iron and zinc are complex with other food constituents, and type of bonds associated could be different for iron and zinc. Boiling beetles significantly reduced the percentage mineral bioaccessibility of iron, sulfur, and zinc. Boiling and roasting had no significant effect on the percentage mineral bioaccessibility of calcium, magnesium, manganese, and phosphorus of beetles. For both insect species, roasted samples had a lower significantly different zinc bioaccessibility compared to raw samples. For all minerals, bioaccessibility of both crickets was significantly lower than a raw samples. Finally, we come to the conclusions. The insect species are a good source of digestible protein as shown by the comparable value of digestibility with that of whey. Boiling resulted in protein loss for both insects, while roasting did not. Boiling resulted in a reduced protein digestibility in both insects, whilst roasting had no significant difference for beetles. However, boiling duration had no significant influence on protein digestibility for both species. Boiling and roasting didn't have any significant influence on the iron and zinc content for both insects. Beetles are a better source of bioaccessible iron than cricket, an indication that mineral bioaccessibility in insect species dependent. Therefore, concerning promoting edible insects for nutritional security because of their high mineral content, each species needs individual evaluation for bioaccessibility. Overall, protein, iron, and zinc in wild harvested insects can substantially contribute to human nutrition. That's all for me. Thank you for your kindest attention and goodbye.